Welcome back. Yesterday we have seen basics of gene expression. Today let's proceed further. Let's start from FastQ file. So this is a FastQ file which is downloaded from NCBI. Now because it's a zip file, I use the command zcat followed by the file name, and I just want to see the first few lines of the file. I use head. So 4 lines represent one sequence, in which the first line is the name of the sequence. The second line is the actual sequence which has been sequenced by a sequencer, and the third line plus, which indicates after this particular line you get the quality score. What is the quality score which I have been talking about? The quality score is also called the Phred score. So let's get into the details of Phred score. Phred score, when a sequencer calls out a base A, T, G or C, a probability score is assigned to it. That's called the Phred score. The higher the score, the lower the probability it is an error, and the lower the score, the higher the probability is an error. It has a lower confidence. So if a Phred score of 10 means there is a probability that 90% is accurate, 10% it's wrong, and that's how it goes about. So it's impossible to determine the quality of all these sequences manually. So we have a tool called FastQ tool which helps us to determine the quality of the file. Let's run the FastQ command. This is the tool FastQC. It's located in this particular location in our server, and we are passing both the files. Now the FastQ analysis is done on both the files, and we get 2 files as we have discussed yesterday, a zip file and a HTML file. This is the HTML file. This is how it looks like. The basic statistics, and the first parameter is per base sequence quality. What does it determine? Across the length of the sequence for all the bases, what is the average quality? For example, if there are 1 lakh bases, at the first position of each of the 1 lakh bases, what is the average quality? So for 1 lakh bases for the 1 lakh sequences at the first position, the quality is 32 standard. There is no deviation. At the second position of the 1 lakh bases, the average quality is 32. Now at the third position, across the 1 lakh bases, the average quality is deviating from 32 to 38. That's how we read. It's a box plot with whiskers on both the sides. I'll be showing you another examples in the document which we have made. This particular thing can be divided into 3 zones: a red zone, yellow zone and a green zone. The red zone indicates it's of a bad quality sequencing. The yellow zone indicates it's a average quality sequencing, and the green zone indicates it's of a good quality sequencing. Let's see some more examples. How does it look like? So this is a documentation which we have made. So this is the per base sequence quality, the same thing which we have seen now. Across the length, at the first position, the average quality. Second position, average quality. Across all the positions, across all the reads, the quality is maintained good. Everything is from 32 to 38. The Phred score is maintained. Now let's come to the bad quality. If you see here, it's usual that as we go to the 3' end, the quality falls down. But is it okay to fall down so much? No, it is not. So here how do we read this? Most of the reads from position 26, the quality went very bad. It's in the red zone, and in some cases from position 18, the quality has fallen down. That's also not good. It's better to reject this kind of a data, because we have spent some money on it. If we want to make use of this data, if in case, what we are supposed to do? We need to trim, clean, cut off these bad quality portions in the reads. That is done using tools which would be discussed a little later. Okay, this is the per base sequence quality. The next parameter which is interesting is per tile sequence quality. As you know, in a sequencing machine, Illumina, you have a flow cell. The flow cell is divided into small tiles. The sequencing should happen in a uniform way in all these tiles. If it doesn't happen, it's a technical glitch. A complete blue graph indicates all tells are accurate, all tells are proper. It's a good sequencing. But if you see something like this, it indicates that there is a problem in the machine, especially in the flow cell, which it has to be corrected and set it right. The sequences from this particular flow cell have a very bad quality. The sequences from this particular flow cell have a bad quality, similar to this, similar to this, similar to this. That's how this indicates the bad quality score. Now, let's get into our next parameter. Per sequence quality score. What does this mean? What is the average quality of a sequence? Okay. So, this is the quality. You take the first sequence, if the bases are 100, sum of all the average divided by 100. And what is that quality? When you see, the average quality score across the sequences is maintained at 40, which is a very good sequencing. Now, let's see what is bad. So, this is a good sequencing. The average quality score for majority of the sequences is at 40, and this is a bad sequencing which shows 2 peaks. Most of the sequences have a good quality, but there are chunk of about 10,000 to 12,000 sequences whose quality is bad, which indicates might be due to the improper flow cell or improper sequencing handling machines. It could be any of these reasons. That's how this indicates per sequence quality score. Next, per base sequence content. So, at each position, this is typically a very bad quality data, per base sequence content. At each position across the length of the sequence, at the first position, the probability of occurrence of ATGC, the diversion should be or a deviation should be approximately 10% or low. Generally, these should be as parallel lines with some wavy appearance. Such prominent peaks across the lines is not allowed, and in some cases, majority of the times, we see these abrupt peaks only at the first 7 bases. If you are not comfortable with that, we chop off or trim off uniformly for all the reads for these first 7 bases. Okay, now let's get into more details of it. Per base sequence content, this is what we have discussed in our analysis before. This is a good graph where all the lines more or less are parallel, and this is a bad graph which indicates that there is a strong deviation of the bases ATGC across the length in the reads. Now, next one, per sequence GC content. Every sequence, what is the average GC percentage seen? Generally, for humans, it's approximately 40%. So, most of the sequences should have a good peak indicating that there is an average of 40%. But if you see here, it's a bad plot where this is the normal distribution. Here, the plot is very close to the normal distribution. The blue line is the theoretical distribution which is expected, and GC is the predicted one. Overlapping, it's good. But here, if you see, there is a peak to the higher, which is not so good. So, why does we take a GC percentage? For example, humans have 40 to 50% GC content. When you have a strong deviation from it, it indicates that there is a contamination, and this particular peak indicates that some percentage of the reads are having a higher GC percentage than the rest of the reads. For example, if you have more than 2 or 3 peaks, it seriously indicates a contamination, bacterial contamination or any other organism species contamination other than humans. That is per sequence GC content. Next, per base N content. Now, why do we do sequencing? To determine which base is present in the DNA or an RNA of our interest. For example, after you do sequencing, all the bases have N instead of ATGC. It's better if we don't do sequencing. The number of N's in a sequence should be minimum or low or no. Okay. For example, let me show you an example which we have here. ZGAT head. If you see in this particular sequence, the first position of the base is called N, means the sequencing machine was not clear whether to call it A, T, G or C. When it's not clear, it just writes N. For example, if all the sequences have only N, there's no point in doing the sequencing, right? It's a waste data. So the number of N's should be 0 or minimum or to the lowest possible. 0 is preferable. Right? Next, sequence length distribution. Generally, if your data is from one sequencing machine, generally it would be of the same length, 1 to 150 bases. 150 bases if it's an Illumina, 76 bases again if it's an Illumina. So the distribution, the length of the sequences should be the same after you do sequencing, before any cleaning or trimming has happened. Now let's see any good quality or bad quality data. Okay. Of course, we don't have an image here. If you have multiple length sequences, you will have different peaks like this. The next part is sequence duplication levels. So what are sequence duplication levels? Some bits of sequences are duplicated in your sequencing because they might be highly expressed genes, and sometimes the duplication indicates PCR bias over sequencing. In RNA sequencing, it's usual to find the duplication of housekeeping genes or super highly expressed genes, but it's not usual in other techniques. Now, this is a bad graph showing duplication level, and here it gives you some statistics. If the sequences are deduplicated, if the duplicated sequences are removed, only 6.74% of the sequences are retained. It means it's a very bad data. Okay. Next comes to the overrepresented sequences. Sometimes some portions of your sequences are overrepresented. We would be interested in what it matches to. For example, if you copy this particular sequence and go to NCBI BLAST, okay, it's taking some time. Go to nucleotide BLAST and blast this particular sequence. Of course, you have to enter it into a FASTA format, just a header and a start, and just blast it. We'll see why it is what is that overrepresented sequence is. It might take some time, we'll just wait. Yeah, we are interested in humans. It's something from chromosome 7. Let's check to what it's matching to. It's matching to some genomic DNA which has no name. Okay, so that's how we keep checking our overrepresented sequences. Sometimes it might be a chunk of DNA which is highly expressed. That's okay to retain, but if you have more number of overrepresented sequences, it's better to delete them off. This can highlight the presence of adapters or contaminants, or in biology it might be a rRNA contamination or very abundant genes. That's how we talk about overrepresented sequences. Now let's get into the last part, which is adapter content. In sequencing, it is usual to have adapter contamination because adapters are the bits which are required in order to start a sequencing process. So the adapter contamination should be the least or lowest or should not be there. Let's see few cases of a good example and a bad example. This indicates there is no adapter contamination, which is good, and this indicates the adapter contamination is there of what? It's a Illumina universal adapter, and that particular adapter has to be removed from the data. Alright, this is in short about FastQC. If you have any doubts, comment below and we will answer to them. Let's catch up in next few sessions about the downstream steps of RNA sequencing. Bye bye.